Hello everyone, I'm Wilson Good at the Ski Institute and University of Utah, and today I have the honor of presenting to you on my research quantifying the spatial-temporal influence of acute myocardial ischemia on volumetric conduction velocity. Acute myocardial ischemia is a dynamic and complex phenomenon that is known to be highly arrhythmogenic in the early phases. It is a major culprit in sudden cardiac death. Ischemia occurs when the metabolic demand of the cardiac tissue outstrips the nutrients supplied by the coronary vasculature. Previous work by our lab investigated whether or not ischemia forms endocardially, as is traditionally thought, or in a much more distributed fashion, as is displayed here. What we found is that it does indeed form in a distributed manner, which began to lead us to inquire on how this distributed nature of ischemia influences propagation and may lead to the arrhythmogenics we associate with acute ischemia. Cardiac propagation is reasonably understood thanks to the work by Durr and others. However, quantification of conduction velocity in the heart is much more simplistic, typically thinking on the order of the cellular scale, with the fastest conduction velocities along the longitudinal direction of the cell, with a slightly slower speed transverse to that direction. Some models take into account a third direction, a speed propagating into the sheet direction. However, these still do not take into account conduction velocities in the volume, which we do in this study, and during pathologies, such as acute myocardial ischemia. This study seeks to quantify volumetric conduction velocity in the intact heart. We will make these measurements before and during the induction of ischemia. Doing this will allow us to ask a number of questions regarding the relationship between ischemia and the development of conduction velocity changes. For example, what is the degree at which conduction slows during bouts of ischemia? Does the wavefront change direction or refract in response to ischemia? Does, as has been suggested by researchers previously, conduction speed up in the early phases of ischemia before ultimately slowing, a biphasic response? And finally, what is the spatial-temporal relationship between ischemic stress and conduction velocity changes over a bout of ischemia? We set out to address these questions using a combination of high-resolution, three-dimensional mapping of the heart using controlled ischemia and signal processing methods to quantify conduction velocity in the volume of an intact heart. The methodology of this study is complex, but we will walk through the methods in three major steps. First, the experimental setup where we induce ischemia, then the processing of the experimental recordings that allowed us to quantify ischemic stress and activation times on electrodes, and finally, how we created spatial maps of conduction velocity and ischemic stress over time. During our experimental preparations of canine models, we expose the heart and place an occluder around the LAD, as indicated here by the red ring. And we induce a graded ischemic response for about 8 to 15 minutes by varying the load placed on the heart via control of the heart rate and degree of occlusion. For this study, we performed this on about 4 hearts, totaling about 20 episodes of ischemia. The electrical response we measured using these intramural plunge needles over here, capable of measuring electrical data across the wall of the heart with 10 equally spaced electrodes along the shaft with about 20 to 30 needles per experiment. The electrodes are about 2 millimeters apart, when adjacent needles are about 5 to 20 millimeters apart. We can examine what the signals look like from a single episode of ischemia using this color encoding on each of the sample recordings. Each one of these shows a beat recorded on a single electrode, with early beats captured in purple, traversing to later beats captured in the ischemic episode in red. And what we see here is there is quite a diverse response to ischemia, even measured in the same region of the heart during the same episode of ischemia. The signals captured from these experiments are processed in an open source software we developed and shared called Pfeiffer, which allows us to easily isolate a representative beat, baseline correct and filter, and fiducialize the 1,000 channels of electrograms we captured during these experiments. Post-experiment, we excise and fix the heart before creating a subject-specific model with the intramural needles registered to their experimental positions. Using these needles, we create what we refer to as the needle envelope, here shown dotted with a dotted black line. By taking the convex hole of the needle electrodes out of the cardiac mesh and removing the blood pools so that we are left with only the myocardium, you can see the blood pools cut out of the mesh here, we also applied a universal coordinate system provided in the CARP simulation suite, and specifically used the row coordinate to detail the transmurality within the needle envelope. It is within this needle envelope we quantify ischemic stress and conduction velocity. We defined the endocardium as a third, mid-myocardium a third, and the epicardium as a third of this transmurality. In order to quantify ischemic stress, we determined the ST40 potential for every electrogram through the ischemic episode. ST40% was determined by looking at the potential of 40% in the ST segment. We can then track that value over time in one of these rainbow plots, creating a time series that we refer to as a run metric curve. This run metric curve details the temporal evolution of ischemic stress at this electrode position. In order to calculate conduction velocity, we must first identify the activation time measured on each electrode. We do this using the standard min-DVD approach, also implemented on the Pfeiffer platform. We perform this for each electrode in the heart, here indicated by the red line on each signal along a needle. We can take those activation times measured along the needle and represent them as color, here traversing from dark blue to a yellow type color, to help with visualization. And if we look at several of these needles distributed throughout the needle envelope, which remember is a volume of tissue so that we have more needles here in the back, using the activation times measured on each of these electrodes, we can recreate the activation sequence. So using a thin plate spline radial basis function to interpolate the activation times on each electrogram to each node of the needle envelope. We do the same interpolation with the ST40% values as well. To see what the activation times look like volumetrically, here's one of our image-based models of the heart. We can measure the activation times on the electrodes again here represented with color. We can then recreate the activation sequence volumetrically. Let's observe that now. To demonstrate how we then calculate conduction velocity, let's look at a small portion of the needle envelope and look at the underlying mesh structure. These meshes have a mesh resolution of 650 microns, fine enough to capture the details of activation. From the previous step, we have activation times interpolated from measurements for each node. In order to calculate conduction velocity, we take the element-wise gradient of activation times to first estimate the local wavefront directionality, and then estimate speed by taking the partial derivative of the inverse gradient and summing those. This gives us an estimation of conduction velocity for each tetrahedral element in the mesh. In order to quantify the wavefront directionality, we refer to as wavefront refraction, let us examine a small portion of the myocardial mesh. We can observe and estimate the directionality of the propagation sequence before ischemia is induced, and then the directionality for each beat captured during the ischemic episode. And we can estimate the angle between the vectors to get an element-wise estimation of the wave refraction using the dot product. Here we can see the conduction velocity before the induction of ischemia. Note the particularly high conduction velocity on the endocardium, in places as high as 250 centimeters per second, much higher than the 50 to 60 centimeters per second we associate with myocytes. Examining the wave refraction and ST40 at this early time point in the episode does not show much, as we have yet to see the electrical consequences of ischemia. Looking at a time point 2 minutes further in the episode, we see very small changes in conduction velocity and refraction, but the first signs of ischemic stress, showing the typical distributed nature we associate with this acute phase. 4 minutes into the episode, we are starting to see some areas of conduction velocity change, and some refraction as high as 70 degrees, but the changes consist of a very few focused regions of intense change. Meanwhile, the ischemic stress has intensified. 6 minutes into the ischemic intervention, we are starting to observe very large changes to the propagation sequence, with some regions in the mid-myocardium and epicardium conducting close to 0 centimeters per second, and some regions near the epicardium refracting nearly 180 degrees, meaning the wavefront has completely flipped orientation from its direction before ischemia. The ischemic stress has continued to advance, and the disparate regions are starting to form contiguous bodies of elevated ischemic stress. Advancing another 2 minutes to the end of the ischemic episode, there are significant changes to the conduction velocity, with many of those regions we associate with the Purkinje myocyte junctions conducting very slowly, with a great deal of refraction throughout the envelope. Let us examine the distribution of speeds throughout the needle envelope during that same episode. At the beginning of the ischemic episode, the speed global distribution shows that the bulk of the envelope is propagating at around 50 centimeters per second. But there is a sizable portion of the needle envelope that's propagating at speeds faster than that. Looking at the stratified results, we have bin the speeds based on transmural extent. We found that regions of extremely fast propagation were mostly confined to the endocardium, shown here in green, and some in the mid-myocardium, orange. The heterogeneity in speed transmurally was a surprising result. 4 minutes into the episode, we observe global distribution has indeed slowed, but there is an interesting disparity in the changes observed endocardially versus what we see throughout the rest of the wall. The endocardium appears to have, in some regions, have sped up and slowed in others, making the speeds endocardially more homogeneous. We will get into this biphasic response shortly. The speed distribution of the mid-myocardium and epicardium has universally slowed. At the end of the episode, we see that conduction speed across the wall has universally slowed, with nearly all regions of the envelope with speeds less than 50 centimeters per second. Looking at the values of ischemic stress prior to the induction of ischemia, here indicated with the green arrows, we can see that, as expected, there is little ischemic stress present, nor is there much variability across the wall for the 20 episodes we examined, each point showing the median value in the envelope. Looking at the peak of the episode, the ischemic stress does increase as we would expect. However, there is no transmural preference for the development of ischemic stress, supporting the distributed model of ischemia. We see a different story with the conduction velocity across the wall. Initially, the speed is very heterogeneous across the wall with the fastest speeds endocardially, followed by the mid-myocardium and finally the epicardium, all statistically significant. At the peak of ischemia, the speeds become much more homogeneous with no significance observed across the wall. One of the more surprising results we observed was a biphasic response in conduction speed, meaning it sped up in the early phases before ultimately slowing. More interestingly, this was most prominent in the endocardium, with the majority of episodes in this study exhibiting a medium speed up of 2 to 15 centimeters per second before ultimately slowing 10 to 60 centimeters per second in the endocardium. Past researchers have suggested this is likely due to the accumulation of extracellular potassium increasing the excitability of sodium channels initially before they subsequently shut down at the higher concentrations. If we observe the distribution of speed changes relative to the pre-ischemia values at a few points throughout the episode, we can see how these changes are very temporally complex. Very early in the experiment, we see very little changes in speed. However, less than 2 minutes into the ischemic episode, we see very little slowing in conduction, but some significant acceleration in the endocardium, and some mid-myocardially. Note the x-axes here are changing at each frame. 3 minutes into the episode, we see both acceleration and deceleration occurring in the endocardium, with deceleration happening elsewhere. 5 minutes into the intervention, we see almost exclusively slowing endo and mid-myocardially, but a paradoxical acceleration in the epicardium before subsequently slowing a few minutes later. At the end of the episode, there is significant slowing throughout the envelope, with some regions slowing more than 100 centimeters per second. It is clear that the changes to conduction speed in the presence of ischemic stress is a spatial-temporally complex and dynamic. Past researchers have associated this biphasic response to be predominantly along the longitudinal direction, which may explain why we see it endocardially. This is possibly explained by the wavefront endocardially being highly susceptible to local fiber orientation, but as the volume of tissue depolarized grows, the wavefront becomes increasingly agnostic to the underlying fiber orientation, and thus the biphasic response is masked. In summary, with this study we showed that during episodes of graded ischemic stress, we see significant conduction slowing. We observed that ST segment changes typically preceded changes to speed, and we routinely saw slowing beyond regions of heightened ischemic stress. We also observed a surprisingly heterogeneous distribution of speed across the wall that at the peak of ischemia became far more homogeneous. Finally, we observed volumetrically a biphasic response in conduction speed that was most prominent in the endocardium. Making conduction speeds volumetrically in the context of ischemia is an important first step in understanding the arrhythmogenic vulnerability of acute myocardial ischemia, and future work will look at these changes before and during the initiation of arrhythmias. I would like to thank the Computational Electrocardiology Group for their assistance during experiments, model generation, and guidance, the Medical University of Graz for the UVC coordinates, and finally the CVRTI for assisting with the experimental work presented here. So much for listening to my talk. Please let me know if you have any questions. You can also email me at wgood@ski.utah.edu or connect with me on Twitter at WilsonWGood.